Hi, my name is Samantha Lumley. I am in Bio 225. It is May 12th, 2019. I have this small handwritten note just to keep myself on task. And this video is the first video, and it is about our first lab. And the first lab is laboratory preparation. And I found it really helpful in figuring out how I was going to schedule my life and schedule experiments because we have multiple experiments or labs due each week, and some of them require multiple steps. For example, next week we have the first lab is aseptic technique, and the second lab is bacterial enumeration. And each one of those has a culturing 48 to 72 hours and an incubation period of 48 to 72 hours. So if I had waited to do one lab and then did the next lab, I would have been late because it would take each one almost 6 days to complete the lab. So I am glad that we needed to go over the lab experiments, and it forced me to read through each one and understand the process and be able to plan that. For example, the bacterial enumeration has a 12-hour period where you have to let it the broth absorb before you can flip the plate over. And so I have to plan when I do the broth to where I can allow 12 hours, then flip the plate over, and then do culturing for 36 hours, and or after that, then do the culture 36 hours, or the incubation 36 hours later. So it was really good to have to read through that. The next important thing that I learned from this lab was the importance of temperature, light, and airflow on the culturing and incubation period. So in the lab, we had to choose, go through 4 different places for incubation and decide which one would be the best one. And I decided that those cupboards over there would be the best place. The degrees are 24 degrees Celsius. There is no airflow and there is no direct light there. And there's no, the pets and the children can't reach up there. So it's away from everybody. And what I learned from the temperature is that if it is less than 20 degrees, it can take longer for the incubation period. So that would really kind of mess up my timing on my experiments. The next part that we went over on the lab was the importance of protective gear, labeling on the bottom of the agar, or on the bottom of the plates versus on the lid, and the clean workspace, using the 10% bleach solution to clean your workspace. Washing your hands before you put the gloves on and after you take your gloves off. It also went over a portion of what normal flora is, and normal flora is bacteria that's on our bodies normally, and with a healthy individual, it doesn't do anything to us. It actually can help you. But with an unhealthy individual, such as immune-compromised people, it can really cause havoc in their bodies. And the 2 different kind we went over were I have a hard time saying this one. It's easier just to say E. coli because everybody knows E. coli, and that's in our intestines. It can be in contaminated food, and if you eat that, it can make you really sick. What I didn't know was that there's a few different strands of E. coli, and they can cause kidney problems, UTIs, bleeding, internal bleeding, all sorts of really bad things. And I didn't really know about those strains of E. coli. The other normal flora was the Staphylococcus epidermidis, which is, as it says, it's on our skin, on our back. It's likes to be in sweaty places like our armpits. And in a normal individual, it doesn't do anything to us. But if you're immune compromised, then it can really cause. It's one of the leading causes of nosocomial infections, like those infections that people get when they're in the hospital. And it can cause all sorts of issues with their body. The Staphylococcus epidermidis also keeps the Staphylococcus aureus at bay, so that's how it helps our body. And the E. coli helps our digestive system. It helps digest things in our body. So both those things are helpful but harmful if you have an immune compromised system. So that is my video for this lab. Thank you very much, and I look forward to the rest of the semester.